My name is Mike Dyer, and I would like to welcome you to the Science of Childhood Cancer seminar series hosted by the St. Jude Children's Research Hospital Comprehensive Cancer Center. This seminar series just started last week, but there are already 2,300 registrants from around the world. For those of you who just joined, my name is Mike Dyer, and I am the co-leader of the solid tumor program here at St. Jude with Dr. Alberto Papo. Today's seminar is focused on epigenetics in pediatric cancer, and it is a pleasure and an honor to introduce Dr. Charlie Roberts. Dr. Roberts earned his MD and PhD degrees from the University of Washington, where he did his dissertation research with Stanley Korsmeyer. He then went on to internship, residency, and pediatric oncology fellowship in Boston at Boston Children's and Dana-Farber Cancer Institute. He trained under Stuart Orkin and joined the faculty at Harvard Medical School in 2000. And then he then came to St. Jude Children's Research Hospital as the Cancer Center Director, Executive Vice President, and Director of the Division of Molecular Oncology. Charlie's lab studies the role of chromatin remodeling complexes in pediatric cancer, and he will share exciting new discoveries in this area today. I have a tremendous amount of respect for Charlie as a leader and a researcher, and I've been particularly impressed with the way he thinks deeply about fundamental molecular and cellular mechanisms of epigenetic processes in cancer while keeping his patients in mind. Charlie seamlessly integrates basic, translational, and clinical research, and he's a perfect example of a physician scientist. Thank you, Charlie, for taking time out of your busy schedule to share your research with us today.
Thanks, Mike. Appreciate the introduction. When we launched this series, it was not my intent to actually be speaking in it, but Mike invited me from the solid tumor program, and so I'm delighted to present my research today, research from my laboratory. So let me first mention I have no financial disclosures and won't discuss any off-label use of approved drugs. So the work that I'm interested in and that my lab works on actually goes back to the time when I was a postdoctoral fellow in Stu Orkin's lab in Boston at Harvard and thinking about what to work on as a postdoc. And there was a paper in Nature at that time from Olivier Delatre's group, shown here, that reported inactivating mutations of a gene called SNF5 INI1, or now known as SMARCB1, present in the large majority of cases of a very aggressive type of pediatric cancer called malignant rhabdoid tumor. And this caught my attention for a couple of reasons. First, it was the first known link between an ATP-dependent chromatin remodeling complex and cancer. So maybe new ways to understand cancer, maybe new ways to think about therapeutic interventions. But it was also of interest to me because history has taught us that the genes that are mutated in the earliest onset pediatric cancers often have broad roles in cancer and can teach us fundamental lessons. And so here I think about retinoblastoma. It's a rare pediatric cancer of the eye, but it's the disease that fundamentally taught us about the role of the RB protein in tumor suppressors more broadly. And so decided to begin working on this as a postdoc. So first, what is this tumor, this cancer, rhabdoid tumors? Well, it's a relatively rare but highly aggressive cancer of early childhood. It can occur in kidney, brain, but really in soft tissues throughout the body. There are, in nearly all cases, biallelic loss of function of this gene SMARCB1, but all four names are actually in use in the literature, SNF5, BAF47, and INI1, that are present in nearly all cases. So it's a very homogeneous genetic background. And so what is this complex that the gene was mutant in? Because it was actually discovered quite a few years before, actually in yeast. And it's evolutionarily conserved from yeast all the way to humans. And so shown here is two versions of the complex, either the BAF or PBAF complex. And what it does is it binds to nucleosomes. So these are what DNA are wrapped around. Nucleosomes is how DNA is stored. And it uses the energy of ATP to slide them or eject them. And so we know that this was what was happening in cells in culture, but what was actually happening in living cells was unknown. The other thing that was interesting is just the degree of variability of these complexes. In green are the subunits that we sort of considered core subunits. But there are others like BAF45 that's got 4 different genes, A, B, C, and D, each a little bit different. BAF60, A, B, C, each a little bit different. BAF53, A and B, 2 different ATPase subunits. And then whole subcomplexes can be switched out. And so there may actually be hundreds or potentially even thousands of variants of this chromatin binding complex. And so thinking about what is it doing and what's its role. And so one of the first questions I decided to ask as a postdoc was, is this really a tumor suppressor? And can we generate models with which we can study the gene and begin to understand its role? And so at that point, used knockout targeting technology and made mice that lacked SMARCB1 SNF5. So if we made mice that homozygously lost both alleles, those mice underwent early embryonic lethality and were never born. So we made the heterozygous mice, and as those mice were born, they began to develop cancer as they aged. So then to bypass the embryonic lethality, we made mice in which we could turn off both alleles of SMARCB1 by just inducing in the mice. And so here's what happened. The mice did fine for about 4 weeks, and then they rapidly developed cancer. In 100% of the cases, these mice died of cancer. And the median onset of only 11 weeks was remarkably rapid for a single gene. In comparison, P53 inactivation takes twice as long, it would be out here, and the combination of P53 deficiency with RB haploinsufficiency is about 17 weeks here. And so the 11-week median latency was remarkably rapid and suggested a quite potent role in tumor suppression. So I mentioned that we were interested in sort of speculating as I began that maybe this would teach us broad lessons in cancer. For the first few years I was working on it, people had looked for mutations of SMARCB1s in other cancer and just didn't find it. And so it wasn't clear that that bet was going to be right. Until 2010, when the cancer genome sequencing data began to emerge. And so here was this paper from a Delatra's group that I showed you. There was one other hint from Ramon Parsons' group in 2008. And then in 2010, the floodgates opened with the finding that the ARID1A subunit was mutant in 50% of ovarian clear cell carcinomas. And now I just scroll through these quite rapidly. PBRM1 mutant kidney cancer, bladder cancer, hepatocellular cancer, gastric, pancreatic, pediatric cancers, medulloblastomas, breast cancers, Burkitt, neuroblastoma. It just sort of goes on and on and on. And so we now know that this complex is quite widely mutated in cancer. There are 9 different subunits of the complex that are recurrently mutated in cancer and are collectively found in 25% of all cancers, making it the most frequently mutated chromatin remodeler, sort of epigenetic regulator, and begin to putting it on par with the frequency of RB, P53, and others. And so it really then emerged as a major tumor suppressor. Except it was also quite new. The other genes that are mutant at this frequency had generally been studied for up to 3-4 decades. But the recognition of the role of switch SNF in cancer, other than the Delatra paper, the widespread recognition is really only now about a decade old. So we know a lot less about this complex, what it is doing in cancer, both pediatric and adult. And so that brings us to areas of interest for my research lab, and I think really much more broadly for the field as we try and understand how can something that is so frequently mutated in cancer actually drive cancer, and what can we think about it from a therapeutic standpoint. So those questions are, what is the normal function of the switch SNF or BAF chromatin remodeling complex? What's its day job? What are the consequences of the cancer-associated mutations that occur in these subunits? And if one is a geneticist, these are really the same question, because as you begin to study a gene, you often will turn it off in a model system to see what happens. And here we're actually informed by nature as what genes and what particular mutations cause disease and may be of interest. And then, because as Mike mentioned, I'm a pediatric oncologist in my background, always keeping in mind is what dependencies or therapeutic vulnerabilities might result from mutations of switch-NIF subunits. And so if we turn to the first question about what is it doing and how does this alteration of this relate to cancer, one of the first questions we asked is, what's the mechanism by which SMARCB1 loss drives cancer? We have to account for very rapid onset cancer. What's going on? So is it genetic or is it transcriptional or epigenetic, if you will? And our bet was, in order to get 100% cancer onset in 11 weeks, this must be involved in DNA repair and scrambling the genome very quickly and leading to cancer onset. And so we looked at this with Elizabeth McKenna, who was in the lab, a graduate student at that point. She's now executive editor of Cancer Discovery, and postdocs. And we looked at this, and so when we first looked, we found no evidence for substantial change in DNA mutation rate when we mutated SMARCB1. And so we then went to take a look at the genomic landscape of SMARCB1 mutant cancers, work led by Ryan Lee when he was a grad student in the lab. And what we found is that these cancers were very genomically simple. They were diploid, and they really lacked amplifications and deletions. Here's a tumor, so across every row is a cancer. Here's one that has an amplification and a deletion. But by and large, as you go across rows, there was nothing going on other than at 22q11, where SMARCB1 itself is being lost. And this can be anything from a whole chromosome arm loss event to just an intergenic inactivation of SMARCB1. And so what we see, really simple cancers. And in fact, at that point, I was in Boston, and so we went over to the Broad Institute and sequenced these cancers. And at that point, they had the lowest mutation rate for a cancer that had ever been detected at the Broad Institute. And so when I was in graduate school, these on the right are the cancers that I learned about. Very high mutation rates: adult cancers, melanoma, lung cancer, esophageal, and bladder cancer. But it turns out we now know that these are cancers that are exposed to the environment. So melanoma, UV light, and in fact, you actually see the mutation signature for UV there. Lung cancer, smoking and inhaled toxins, GI tract. And so if you move into tumors in adults that are located deep in the body, the mutation rate goes down by almost a log-fold. And in here, you can then begin to start to see things like pediatric cancer, neuroblastoma, but all the way over here were the rhabdoid tumors. And so the exomes had a total of 4 base pair changes on average, one of which was SMARCB1, and the 3 others were just random and seemed non-significant. And so that really brought up an important question. What accounts for the extreme paucity of mutations? So we have a gene that, when mutant, single gene leads to extremely rapid cancer onset, and yet the genomes that result are very simple. So how do we resolve this? And I think a lot about this because there was separate work from Bob Weinberg's lab. They had taken fibroblasts and tried to figure out how many mutations were required. And they would add oncogenes and knock out tumor suppressors, and they came to the conclusion that 5 mutations was the minimum. And Bert Vogelstein's lab separately did mathematics of how many cells in the human body, rate of DNA mutation, incidence of cancer, and they separated with the conclusion that there were 5 mutations minimum. And yet we're seeing something here that is very different. And so how can we resolve that? And so one possibility would be that there are mutations outside the exome at a high rate. So we've begun to look, haven't seen much. It's a big place to look, so we can't rule it out, but we don't see mutation rates that would begin to explain how you get such fast cancer onset. There could be germline events that we're missing. It could be that only certain people that have SMARCB1 mutations get cancer. We don't think this is right because Mendelian inheritance seems to happen, and in mice we put it in any genomic strain and they get cancer. But developmental stage or epigenetic state might be important here. And so what do I mean by that? And so we turn back to our mouse model, here the conditional mice, and what we did, and so I should mention that the mice in here are getting a lot of lymphomas, and out here rhabdoid tumors. And they get a very specific type of lymphoma called a mature T-cell lymphoma. And so we decided to use genetic engineering technology to turn off SMARCB1s at different points in T cell development and see what would happen. Was there a cancer stem cell? So we turned it off here in common lymphocytes to give rise to both B cells and T cells. We turned it off with LCK-CRE in the thymus in early thymic development, and we turned it off in mature T cells. And what we found was actually quite surprising to us, and that SMARCB1 deficient lymphomas arose exclusively from CD8 positive T cells. And in fact, it was even more specific than that. It was a very specific subset of CD8 positive memory T cells. And to me, the corollary here is even more important. The CD4 cells never transformed and gave rise to cancer. The thymocytes in mice, they never transformed, even though mice can very easily get thymomas. The loss of SMARCB1 did not lead to thymomas. And this loss in the B lineage and T lineage, we never saw B cell disease. And so what we found was that the developmental stage or epigenetic state was incredibly important. In many ways, this is just another word for cell of origin. So despite the fact that SMARCB1 loss leads to really rapid cancer onset with very high penetrance, it turns out it's only coming from really specific cell types. And so if we go back to this and think about it, the data from Weinberg was using fibroblast as a model. Well, maybe fibroblasts are actually not a good model of cancer. And the calculations done by Vogelstein's group was taking, at that point, thinking of every cell in the human body as being identical and having the same risk. But if that's not the case, if there's only very specific cell types or very specific developmental windows, then the math goes out the window and new calculations are needed, and a much smaller number may in fact be able to drive cancer. And so this small number of mutations that we find with very rapid onset suggests that the cancer-driving effect of SMARCB1 loss may be largely transcriptional in nature, not DNA damage, but changes in transcription. And if we think about normal development, maybe this shouldn't be such a surprise, because in normal development, we have nerve cells, for instance, that are spinal neurons that are a meter long, don't divide for decades, and have exquisitely specific ultrastructure. And then we also have things like activated lymphocytes that are small round blue cells that are dividing every 6 hours. And for all intents and purposes, these cells have identical DNA content, but they have dramatically different fate. And that's where chromatin remodeling or epigenetic regulation, if you will, may come into power. And we began to then think about this. I also want to point out that this really has some, I think, pretty strong implications for therapy as we think about translating from the lab. You know, as I was sort of finishing training and thinking about things, this was the paradigm, CML, right? Imatinib. If you can identify the target, BCR-ABL, the chemists are good enough, it can figure it out. Good enough meaning that there are tough things to target. This is favorable. You can develop a drug, you can shut it off and have a dramatic impact on the tumor. But what about rhabdoid tumors now? There's only one gene that is recurrently mutated and it's gone. It's not there. So how do you target that? And when we think about this mechanism, it's applying not only to rhabdoid tumors, but this complex is mutated in close to 25% of all cancers. So how do we think about that? And so that then begins to bring into additional questions of what's its mechanism and can we use this to think about therapeutic insights? So what is the function of the switch-NIF complex and what are the consequences of these cancer-associated mutations? And so looking at this, we first began to ask about regulation of transcription. And a couple of years ago, we published these 3 papers essentially at the same time. It really implicated this complex as a regulator of enhancer function. So what are enhancers? And so when I was in graduate school, we learned about genes and that there was a promoter, and the promoter was really what determined whether a gene was on or off. So it turns out that that's actually not the case. And that genes that are dynamically regulated have what are known as enhancers. And depending on how the gene and the DNA is looped, the promoter can be linked up with an enhancer. And that particular enhancer is what determines where the genes are turned on and off. So out where we used to think of as junk DNA is now these enhancers that are actually the instructions, if you will, in a Lego set. They're the instructions that tell where the bricks and everything should be built and when. And so this is work that was led by Xiaofeng Wang, who's a postdoc in the lab, now has his own lab at Dartmouth, and work led by Barack and Kimberly from the lab as well, and from Peter Park's lab. And so to go at this, what we were asking is, if we look at two of the families that we knew about at the time, and here's this gene SNF5 that's also known as SMARCB1 or INI1, and we can turn it off. And so we can inactivate it widely. And what happens when we turn this gene off? And so we were looking at chromatin regulation, and there are modifications on chromatin, in this case, one called histone 3 lysine 27 acetylation, that actually begin to be part of the code that's telling genes when to turn on and off. And so all of these on the left-hand side are control. On the right-hand side are the experimental. We had either wild-type cells, ARID1A, so one subunit of the switch-NIF complex, another subunit, a third subunit, or control EZH2. And what I'll draw your attention to is when we turned them off in all three cases, what we saw was a change here in this H3K27 acetyl that we know is located at, particularly at enhancers and at promoters of genes. So a dramatic change picked up even at the level of the whole cell Western blot. And so what was going on? We then began to ask, was this telling us about a role for the switch-NIF complex in being targeted to promoters, being targeted to enhancers? And so we began to take a look. What's the consequence of this mutation? And so what this is showing is, where is the complex targeted with and without SMARCB1? And so if we look at TSS proximal, this is the name for promoters, what we find is that the complex, whether or not SMARCB1 is there or not, it's targeted to promoters and it's unaffected in a small level. But if we look at these distal sites that are the enhancers, what we found is that in the absence of SMARCB1, there was very little binding to enhancers, and it was the addition of SMARCB1 that now led to targeting of the complex to enhancers. So we're seeing very strong binding that's dependent upon SMARCB1 of the complex to enhancers. And so what's happening then to the chromatin landscape and to enhancer function? And how are we beginning to understand this effect on normal development? And how are we beginning to understand this in disease? Might this be interesting for therapy? So we went forward and took a look. And so these are what is known as ChIP-seq tracks, and we ask what happens. And so these are two other subunits of the switch-NIF complex, and without SMARCB1, they barely bind. With SMARCB1 in brown, we see there's good binding. And if we look at the histone marks, this one and this one are specifically bound at enhancers, and they go very low in the absence of SMARCB1, whereas the promoter-associated mark is really low to begin with, where SMARCB1 is bound and lowly affected. If we take a look at what else is happening, so it's not just about this K27 acetyl. All of these proteins are things that are involved in regulation. The mediator complex, BRD4 complex, CBP P300, this is the K27 acetyl enhancers. And so if we take a cell line where a mutant or rhabdoid tumor cell line where SMARCB1 is off and we add it back, we see that lots of changes are happening at enhancer. The mediator complex goes up, BRD4, CBP, all of these things are going up in two different, whether it be a kidney cancer line or a brain tumor cancer line with rhabdoid. And so now seeing a very broad effect upon enhancers. And so what's happening to transcription when this is happening? And so if we take a look here, what we see is that the genes that are being affected are really things that are involved in development. So we see development, morphogenesis all the way through. And so we see a very strong signal. So we're also trying to understand, so that's about rhabdoid tumor, but what's happening more broadly, because the switch-NIF complex is mutant widely in cancer. And so here's Radhika, grad student in the lab at Harvard at the time. She's now doing her postdoc, looking at the ARID1A subunit. And so now here's the ARID1A subunit. It's only found in the BAF family. It's not found in the PBAF family. And so what happens when we knock it out? And ARID1A is of interest because it's the most frequently mutated subunit of the switch-NIF complex, widely found in many types of cancer. So if we turn off ARID1A, here's what we see. The mice do fine for a while, but then they also rapidly begin to succumb. Now, the SMART-B1 rate of onset was way back here at about 11 weeks, 77 days. The ARID1A takes longer, but then they begin to rapidly develop. And these mice, now they're not developing rhabdoid tumors, they develop colon cancers. And so they get very aggressive invasive colon cancers. And all the models that had generally been used up until that point would not really recapitulate human colon cancer very well. They would get small intestinal polyps, and then you wait long enough and those mice would get cancer. In the case of the ARID1A, they never developed these polyps. They only developed invasive cancer, and only in the colon, not in the small intestine. So a much better match for the human disease. So what's the effect of ARID1A on the chromatin landscape and upon enhancers and enhancer function? We see something very similar, just a little bit less strong than loss of SMART-B1 alone because of other residual complexes. We see K27 acetyl go down, and now in the case of K4 mono, not as strong an effect, although this nucleosome-free region where the transcription factors normally bind is in fact wiped out. And so if we look at the enhancer conclusion, I'll pause in just a minute and take questions as I finish this slide out. So if there are questions, and Mike, if you want to read any. So the enhancer conclusion is that the switch-NIF complex turns out to have a major role in the facilitation of the function of enhancers, and that this underlies the ability of cells to differentiate. And so as we see the functions, even typical enhancers and super enhancers get affected. And interestingly, unlike BRD4 that Jay Bradner published that most strongly affects super enhancers, here we were finding a particularly strong effect upon typical enhancers and impairing their function. So let me pause there midstream and see if there's any questions, and after those I'll pick up and keep going. Mike?
Yeah, we have a couple of questions here, Charlie. So the first is long non-coding RNAs have been shown to play an important role at enhancers. Have you looked into the role of non-coding RNAs in targeting the BAF complex to specific enhancers?
So it's such a great question. There has been a lot of work that's begun to look at what is bound, in fact, the long non-coding RNAs, and a number have been found to bind to the switch-NIF complex. John Prenzner, when he was at Michigan, looked at some of this and found one that specifically binds and interacts with SMART-B1. We're also now learning more and more about enhancer RNAs and those, in fact, being strongly built. In my lab, we have not yet taken a look at these, but I think we're going to be finding increasingly the polycomb complex has had a number of these things. And so this may well be sort of a hidden piece that is underlying targeting and still to be determined.
All right. Great questions. There's lots coming in, so give me 1 sec. All right. Do you see similarly low mutation burden in rhabdoid tumors and pediatric ALL, or even less?
Yeah, so great question. So in MLL mutant pediatric leukemias, which strike infants and toddlers, they're also very low. So right in the same range as this. Interestingly, so some adult cancers that have switch-NIF mutations have very high mutation rates. And then we're trying to figure that out. So it turns out that ovarian cancers, where people have a germline BRG1 switch-NIF mutation, those have very low. So what we think is happening is the switch-NIF mutation's very low, but you can get other DNA damaging events and then lose a switch-NIF, and some of those cancers are complex.
Are the affected enhancers related to known oncogenes, stemness genes? Is there sort of a category that jumps out at you?
Great question. In fact, this is exactly what we were focusing on and thinking about so much in the lab, and I'm going to go into that more in the back half and as I bring into the model to bring you actually what I think is, we'll put a nice bow on the mechanism.
Okay, we'll do one more and then we'll keep the rest for at the end. The last question for right now is, in the colon cancer model, do the tumors start to accumulate additional mutations as they progress?
So good question. So mice tumors are, at least when we've looked at them, difficult to figure out whether there's more. We did a few sequencing of a couple of them and didn't pick up much happening there. We should go back. We should probably work with Dr. Dyer and go back and take a look at some of these and see really what is there. Because what's interesting is the colon can develop completely normally. We went back and knocked it out entirely in the colon. Colon develops completely normally without ARID1A. and then the tumors begin to form. And so it's a really interesting question of what else is bringing that along. Okay, so I'm going to continue on, and then we'll have time, as Mike said, we'll have time. I'll save time before the hour's up for questions, and then happy to continue. The talk will end at the hour, but if people are interested in staying on, I'll take additional questions then. So those are some of the mechanisms we were thinking about, and then we wanted to think about also what are therapeutic vulnerabilities and dependencies. And so in many cases, these things have had strong intersectionality. So returning to this question again of therapy and understanding mechanism, what are we possibly going to think about therapeutically for rhabdoid tumor that falls in the realm of targeted therapy? So asking this question, what dependencies result from mutations of switch-NIF subunits? And the first group that we looked at were things that we called mechanistic hypotheses. And so I'm going to draw two distinctions. One is the things that we thought about in lab, read the literature, what might be going on, and a second set of things that we took a relatively unbiased look for. So in the mechanistic group is something called polycomb or EZH2, polycomb repressor complex, writes the PRC2 complex of which EZH2 is a member, writes a silencing mark H3K27 acetyl. And so as we had read through the literature, we found that Drosophila geneticists in the 1990s were looking at polycomb mutant flies. And these are flies that have abnormal body patterning, too many wings, too many legs, not enough legs, patterning defects. And when they did screens for what would suppress a polycomb mutation in a fly, out popped switch-NIF mutations. And so, and to the degree that in flies, switch-NIF is called trithorax group genes or polycomb antagonists. And so we wondered whether this might be relevant to mammals. And Peter Verizer's group had done an interesting study, and we got interested in this. And so what we did is take a look, and now this is all the way back in 2010, but when we knocked out SNF5 or SMARCB1, what we found is at the signature locus of P16-INK4A, that we would get a significant increase in K27 trimethyl. So indicating that polycomb function was now increased. And we asked, is this in fact broadly happening? So if one takes embryonic fibroblasts and deletes EZH2, genes that are K27 trimethyl modified go way up. And so we asked the opposite. We took these SNF5 deficient maps and we turned it off, and we saw the complete opposite. Things that had K27 acetyl K27 trimethyl, associated went down. And so we think that these are poised genes that are dropping. So we used our genetic models to ask, is this play out in vivo? And so remember we had the SMARCB1 mutant mice. Well, now we put EZH2 conditional on top of that. And so here's the SMARCB1 mutation. If we make them additionally EZH2, now it completely wipes out the tumor formation. In fact, all of the CD8 T cells are there, they just never transform. And we tried this in other models. We did an osteosarcoma model, knocking out EZH2 had no effect. And in a prostate cancer model, knocking out EZH2 actually accelerated tumor onset. It was only in the context of the SMARCB1 SNF5 mutation that it completely blocked tumor onset. And so at that point, we published what we called a sort of epigenetic antagonism between the switch-NIF and polycomb complexes, such that SNF5 SMARCB1 was opposing the function of polycomb and regulation of lineage-specific programs. And loss of this control would then underlie oncogenic transformation. And this is the series of papers that we first found this with SMARCB1 and then broadened it more widely to switch-NIF complexes. So we were doing this and we published this, and then a company called Epizyme picked up on this and began testing their EZH2 inhibitor. They were interested in EZH2 because it has a gain-of-function mutation in lymphomas. They found that it worked in rhabdoid tumor cell lines, called us up. I never took money from them, so I independently from them. And then they began to move forward and went into clinical trials, first in Europe and in adult patients with solid tumors, and looked at EZH2 inhibition. And what they found here in this waterfall plot was the responders seemed to have mutations in one of the two switch-NIF subunits. Very nice work then from Sigal Kadosh and Jerry Crabtree's lab found also antagonism. So this is more recent. They did a very nice in vivo editing in living mice, recruited the switch-NIF complex to a specific locus, and found that within seconds it would displace the polycomb complexes. And so very strong evidence of antagonism in transcription. And so there's clear antagonism between SMARCB1 and EZH2 in transcriptional regulation. This results in unchecked activity of EZH2 when SMARCB1 is mutated. And these discoveries led to then additional clinical trials. And we were very excited quite recently, earlier this year, actually FDA approval of the EZH2 inhibitor tazemetostat, specifically for patients who had INI1 loss or SMARCB1 loss. And so this was really a fantastic success for the lab. The first target we identified and went after makes it all the way to FDA approval. Great, and that's fantastic, and I think inspires us going forward. But I think there's also really important complexity not to be lost in this story. And I think it teaches lessons not only about this, but how we have to think about therapeutic targeting and mechanistic pathways. And so we were asking a question, actually, what's the chromatin landscape in human rhabdoid cancers? And this was actually a question that Stefan Pfister approached me at a meeting. We were in Singapore, and he was doing work with Marcel Kuhl. And they began talking to me about this. And so this was really their project. We played a fairly small part in it. But I think really interesting. And that is, so they were doing ChIP-seq for histone marks in primary pediatric brain tumors, in normal brain, and in embryonic stem cells. They basically wanted to say, what's the chromatin landscape in the tumors? And so they'd done this. And so as they began talking to me and then asking the question, I wanted to know, and I was talking to Stefan, how do enhancer domains look in the SMARCB1 deficient rhabdoid tumors? And they were looking at the brain where they're called ATRT. And so exactly as we predicted, as our data would show, here are the brain tumors, the ATRTs, the rhabdoid tumors. The enhancer domains are very low compared to GBM tumors, medulloblastomas, adult normal brain, pediatric normal brain. They're all the way down by embryonic stem cells. So perfectly predictive. So I said, Stefan, take a look at the polycomb domains and how do those compare to the other cancers? Because our prediction should be that those should be up and through the roof. And so what we found was not only not what we predicted, we found exactly the opposite of what we had predicted. The polycomb domains were low. So here's GBM, medulloblastoma. Remember, our prediction is polycomb domains should be high, and they're way down in the basement. Completely the opposite. And so this was very confusing as I heard this. And so I talked to Marcel and to Stefan. I said, do me a favor. Do ChIP-seq for the switch-NIF complex in the normal brain sample so we know where switch-NIF is normally bound. And then let's restrict our analysis just to those places that switch-NIF is bound. And when we did that, now we found exactly what we predicted. That in the rhabdoid tumor cell lines, so the tumors, the K27 acetyl was low, and in the K27 trimethyl, very high in the ATRTs. Okay? So what we found is consistent with our earlier finding. This data indicates that loss of SMARCB1 results in high levels of polycomb binding and K27 trimethylation wherever switch-NIF was bound, so a direct effect. However, secondarily, at many other sites, K27 trimethyl levels plummet. And what we hypothesize is happening here as the cell begins to detect inappropriate and failed repression inappropriate repression of polycomb now with the absence of SMARCB1 and the switch-NIF complex. It tries to down-regulate PRC2, the polycomb repression. It can never get it back to normal where the switch-NIF complex was, but everywhere else in the cell, it now goes low in compensation. But it then becomes very important to think about this because the therapeutic implications of EZH2 inhibition, if this is always happening, then there's going to be some sites that have very high levels of EZH2 and some sites that have very low levels of EZH2. And so ultimately, the impact upon therapy is going to have to be worked out in greater detail in the lab through clinical trials. I'll return to that as well. Okay, so then what other therapeutic vulnerabilities result from this mutation of the switch-NIF subunits? And so now taking these unbiased approaches. So what about things that we're not smart enough to think about in rhabdoid tumor that I have a hypothesis about? And so here, before I left Boston with Kim Stegmeyer and Todd Golub and the Broad Institute, we established what's called the Pediatric Cancer Dependency Map. So putting in pediatric cancer cell lines, characterizing them, doing genetic and chemical perturbations. So initially shRNAs genome-wide, and now we've gone through first the Gecko library and now the Ivana library of near genome-wide CRISPR inactivation, and are now going through chemical perturbation, do computational analyses, and develop therapeutic hypotheses. I really should have updated this slide. I realize it's not, at one point we had 8 rhabdoid tumor cell lines. We're now, I think, close to 15. And there's over 100 pediatric cancer cell lines and over 500 adult cancer cell lines as part of this. So we get really robust data. And so one of the first things that we noticed as a vulnerability was a gene called BRD9. And so here's what it looks like in the waterfall plot. It wasn't the strongest one that we saw, but they were all to the left, and it caught our attention partly for that reason. And we went into the lab, and I won't really show you and validate this, but when we knock down BRD9, we see loss of growth of the rhabdoid tumor cell lines, particularly in rhabdoid, and if we do it in other cancer cell lines, we don't. So why was this of interest? What was BRD9, even though it wasn't the #1 hit? It was of interest because BRD9 is itself a subunit of the switch-NIF complex. And so it had been reported to be. And so we want, is this in fact real? And so we immunoprecipitate BRD9. And bottom line is, and these are rhabdoid tumor cell lines. You indeed pull, all of these are subunits of the switch-NIF complex. The nomenclature is horrible, but these are all subunits of the switch-NIF complex. And then when we do it in wild-type SMARCB1 lines, we saw something very interesting. And that was this. That even when SMARCB1 wasn't mutant, there was no interaction of BRD9 with SMARCB1, despite the fact they're both parts of switch-NIF complexes. And so what it led us to realize that while switch-NIF complexes share ATPase subunits and a number of other subunits, and there were 2 known families that had distinct subunits, the PBAF and the BAF, we discovered that there was another family of complex, the BRD9 containing complexes. And so there wasn't just 2 families, it turns out that there was a 3rd family. And the loss of SMARCB1 was inactivating the BAF and PBAF complex and leaving behind this BRD9. And so this vulnerability screen let us learn about the existence of the BRD9 containing complex. Gave us great new mechanistic insight. And so we were not the only ones to find this BRD9 complex. Emily Doykin had identified this, and Segal Kadosh lab had a very nice paper also finding BRD9 role in assembly, as well as Scott Armstrong's lab. And so what we found was now that BRD9 is functioning separately from the complexes that contain SMARCB1, and I'll show you that in a model as we close out. We're learning more and more now, how are these mechanisms and redundancies working? So we were using an additional mechanism of relatively unbiased taking a look. And this was actually something that was started by Stu Schreiber's lab. Tom Howard and Elaine Overlook were grad students in the lab. And so Cancer Target Discovery and Development Project, this was led by Stu Schreiber's lab at the Broad. And the idea here was to take lots of cancer cell lines, in this case 900 cancer cell lines, or 857, treat them with 481 different drugs or compounds that were either FDA approved or coming along, generate 16-point dose curves for every drug and every cell line, so enormous complexity. And then take a look at the compounds and find out which cancers and which were being particularly killed. And so what happened then was my phone rang when they did this. And so what they found initially was, so here's this drug called amacitaxine, and here are the rhabdoid, which is FDA approved, and here are the rhabdoid tumor cell lines. It was the one, and this is what 900 cancer cell lines looked like graphed in a dose response curve. And so it was the one drug in which all 8 rhabdoid tumor cell lines that we tested ran to the front and were highly sensitive. And so we then compared them to CML, because CML is what is the FDA approval. CML is in red, the rhabdoids in blue, and so they are at least as sensitive as the CML. as we look here. So here's CML, rhabdoid tumors as sensitive as CML, and there's control cancers. So we go in vivo now and take a look at amacitaxine. And so while we treat, completely blocks proliferation while amacitaxine is present. And at the dose that we used, it 100% stopped proliferation. We just did continuous infusion for 28 days. And then stopped infusion at that dose, and then the cell lines would grow back. So it was able to completely give us stasis at the dose that we used, and then they would grow back. So we began to wonder what does a ATP-dependent remodeler have to do with amacitaxine? And so amacitaxine binds RNA polymerase and shuts down translation. So it's translation inhibitor. So it binds and completely shuts down translation. And so we took a look and began to think about what is going on here. And so it has been shown to cause loss of anti-apoptotic MCL1 in rhabdoid tumor cells. Sorry, it was shown in chronic myelogenous leukemia, and we began to wonder then in rhabdoid tumor cells. And so took a look, and indeed, when we see that MCL1 goes down, and these levels go down before cleaved caspase 3 begins to go up. And so we began to then think about short-lived chromatin regulators which influence transcriptional output. And so there was a very nice paper in Cell Stem Cell that shows that chromatin regulators that dynamically regulate tend to be very short-lived and are very much dependent on translation and translation inhibitors that can block. And so we think in mechanistically now, what is happening is in the absence of SMARCB1, there is production of trying to ramp up what little switch-NIF complex is left behind, and this now leaves a very strong dependence upon the translation of those complexes. We're still working on nailing that mechanism. That then begins to understand why we see with impaired translation. So interestingly, when we did a CRISPR screen for resistance, we found exactly that this was an on-target drug and that it was loss of an impaired translation that underlied this. So I want to then talk a little bit about therapeutic mechanisms and some conclusions and then models, and then I can take questions. So it has turned out that as we begin now with something that we have a gene that is a protein that is missing, and we begin to study mechanism, we now, and both mechanism and unbiased screens, begin to find therapy. And so these in the last year are 4 papers that we have published that have come out of these now unbiased screens. In some cases, we have found beautiful mechanism that we understand, as well as, for instance, the BRD9. And in other cases, we're finding things that are very true and going into clinical trials, the MDM2, MDM4, that we're still working on mechanism. So this screen was designed, we wanted to do it to try and get to things that could get us therapy as quickly as possible. And in fact, we're now finding that these are going into clinical trials. In fact, one has already gone and been opened up now at Dana-Farber. We've got another one with a different molecule here at St. Jude coming behind. And so we're really learning about things that, in addition to things that are broadly targeting, we found that certain receptor tyrosine kinases become targets, and it depends upon the cell of origin. So in each of the cases of the rhabdoid tumors, we find a receptor tyrosine kinase being activated by the loss, but it depends on the cell of origin, and so which particular RTK is the dependency here. And then down at our core mechanism in which we think about antagonism with the PRC2 complex. Bringing us insights. And so we're excited about these going forward. We continue to work quite intensively on the mechanistic basis, bringing together the vulnerability basis and linking them. And so a number of the people in the lab that are working on those. So finally, some conclusions, and then a model, and then questions. And so the switch-NIF or BAF complex mutations are very common in cancer. 9 different subunits are mutated, 25% of all cancers. SMARCB1 deficient cancers contain remarkably few genetic mutations, suggesting a transcriptional mechanism. Inactivation of SMARCB1 or ARID1A in mice leads to onset of cancer, but from very different cell types. So remember that the SMARCB1 loss does not give rise to colon cancer, the ARID1A does, and so we see very significant context dependence. The switch-NIF and polycomb complexes have antagonistic roles in transcriptional regulation. And that loss of either SMARCB1 or ARID1A results in loss of active chromatin at enhancers and impairs genes involved in development. We found that BRD9 is a member of a novel family of switch-NIF complexes that lack SMARCB1, so beginning to understand more of this complexity. And in the absence of SMARCB1, BRD9 containing complexes become essential and may be therapeutically targetable. I mentioned just briefly the receptor tyrosine kinases and MDM2/4 that I don't have time to show you. And so if we look at the last two slides now, one is what do we know about how this complex works? And so it turns out that this canonical BAF complex, we and others have now found is bound strongly at enhancers, and this is the one particularly affected by SMARCB1 loss. The PBAF complex is bound largely or predominantly at promoters, and this new complex that we identified and others, the NC-BAF or G-BAF complex, is located just downstream of promoters. And so together we're seeing a cooperative activity of these three complexes working together collaboratively to facilitate gene activation and opposition to polycomb complex. So in the last slide then, what is the model and what's the takeaway by what's really going on here that's driving cancer? So a couple of things to remember. First, only highly select cell types are transformed. Remember, CD8 T cells, whatever rhabdoid progenitor is, whatever the colon cancer progenitor is, and we're not seeing widespread cancer. We think the susceptible cells may have a high proliferative capacity. So memory T cells, they sit there quiescently until they encounter antigen, and then they're highly proliferative. In fact, we showed that antigen is important for these lymphomas to actually form. And so what we think is that it is the enhancers then in these cells that are impaired, that preclude differentiation. So if we think about normal cells and normal development in our body, as we need to replace cells, hematopoietic stem cells, for instance, divide, and then they make enough cells, and what happens? They differentiate, and they make neutrophils or T cells or whatever. And how do the cells differentiate? Well, the transcription factors bind to enhancers, and they turn them on to differentiate. But when the switch-NIF complex is broken, those enhancers can't turn on. And so those signals for differentiation come in, but they're not listened to. And so it's a setup. In most cell types, this is not cancer. It doesn't result in cancer. But if you're a highly proliferative progenitor cell that has just the wrong transcriptional programs on, when the switch-NIF function is lost and an enhancer are lost, it may block differentiation, and cells then become locked in to this highly proliferative state. And so as of August of 2020, we think this is likely the mechanism that is contributing to rapid onset cancer and high prevalence cancer. So I just mentioned the people in the lab, the Roberts lab, and I tried to call out the names of the folks that are working on the projects or had led the projects that I was talking about as we went along. Very important collaboration from Peter Park, computational biology at Harvard Medical School, Ramesh Shivdasani, GI oncology, working on the colon cancer. The Pediatric Dependencies Project at the Broad, if you go to the Broad Institute and just look up Dependencies Project, Dependencies Map, you'll find this. Kim Stegmeyer, Todd Golub, Bill Hahn, Stu Schreiber's group, Matthew Reese, a number of folks at St. Jude, Janet Partridge, Beth Stewart, and a couple of our papers, Mike Dyer, and then funding from NCI, and particularly ALSAC St. Jude in terms of the St. Jude collaboratives that have funded this. So I will stop there, and I'm happy to take questions.
All right, Charlie, beautiful. Thank you so much. So a couple of questions. The first one is a technical one, so we may want to go back to some of the slides if possible. Sure. But it was about the ChIP-seq tracks and asking why are there two peaks when looking at the enhancer ChIP-seq? And so I'm not sure if that's about the two lines or about the nucleosome-free region, but maybe just describing that in detail.
Nucleosome-free region, if I can get back there.
Yeah, it's a ways back.
I know I speak fast, and so I use a lot of slides for going slow. So this peak right here, actually, is what is the nucleosome-free region. And so what happens is transcription factors bind at enhancers, and they're the ones that kick off that gene turning on. In order for them to bind, a nucleosome can't be there. If the nucleosome is there, the transcription can't bind. And so what we think is happening is the switch-NIF complex is coming in, and moving those apart, opening up the nucleosome-free region. That sets off a whole cascade of K27 acetylation and other proteins that are involved in holding this open. And so as you lose switch NIF function, you begin to lose this nucleosome-free region, and now the transcription factors can't bind. And this is what I was talking about, a block in differentiation. The transcription factors are there, they just can't function and get their job done.
Perfect. Next question. We've seen increase in PRC2 in radiation-resistant GBMs. Given the relationship with SMARCB1, have you observed that SMARCB1 mutations lead to increased radioresistance in rhabdoid tumors?
We have not directly looked at radioresistance. I think it's an interesting question to look at. The PRC2 complex in EZH2 tends to be highly expressed in rapidly dividing cells, and so not all cells that express high levels of EZH2 are necessarily dependent on it. At least they haven't shown that way necessarily in drug response. So it becomes an interesting place to take a look and see if it's there. You know, my initial hypothesis would be that it maybe wouldn't be particularly dependent because we don't have what looks to be a DNA damage phenotype. But then there are other very simple cancers, actually beautiful work from Beth Stewart and Mike looking at Ewing sarcoma that find, despite them being quite simple, that there can be vulnerabilities to PARP inhibitors. And so I think this needs to be looked at carefully.
All right, great. Next question. What are the mutations? How can you characterize them for any specific changes in the amino acid composition of these proteins? I think looking at, is it inactivating mutations or any gain of function possibility there?
Great. And so in terms of SMARCB1, these are all loss of function. They're nonsense, they're frame shift, they're deletions, they're gone. The prevalence of mutations appear to be inactivating. In the ATPase subunits, the ones that actually hydrolyze the ATPase and generate the force, there can be point mutations there that are specifically located in the ATPase region. And so those tend to be loss of function, but loss of the ATPase function. And it's not clear whether the rest of the protein is actually having some function, and it may indeed. Then there was also, Segal did some really beautiful work looking in synovial sarcoma, where it actually appears to be a gain of function effect. And so rhabdoid tumor loses SMARCB1, anyone. Look at synovial sarcomas, they also have very low levels of SMARCB1. And so the thought was going to be, aha, these are the same or very similar. And it turns out there's a chromosomal translocation in synovial sarcoma that fuses a switch NIF subunit to another protein that knocks SMARCB1 out. But it turns out the loss of SMARCB1 doesn't seem to be what's driving. And these now actually hyper displace PRC2 instead of losing their function against PRC2, they hyper displace it. And so we again get this context dependence, which I think is very interesting. EZH2 can have loss of function mutations that drive AML and myelodysplasia, or it can have gain of function mutations that drive lymphomas, B-cell lymphomas. And so it was a bit of a long-winded answer, but mostly loss of function, but also some real context specificity that suggests gain of function can also play a role.
Okay, last question for the official portion of the presentation. There were two questions related to proliferation. So the first is a cell of origin for the colon cancer model highly proliferative? And given the high proliferation rate, are these tumors really sensitive to chemotherapeutics that target proliferating cells?
It's a great question. So we don't know the cell of origin for the colon cancer yet. We did something that would knock them out in colon cancer stem cells, so down in the base of the crypts, and so you lose ARID1A throughout. But as you know, we never caught the cancer as it was forming to really know what cell it was coming from, and those cells can migrate up. So we really don't know what the cell of origin is in any sophisticated way. So these tumors are quite rapidly progressive. They tend to, in fact, respond to chemotherapy initially, but they come back quite quickly and tend to be refractory. So these are some of the worst actors in pediatric cancer. So rhabdoid tumors have a very high fatality rate very quickly, despite the fact that they respond to chemo upfront.
Fantastic. Thank you again, Charlie, for taking the time to share your research. This officially ends the presentation, but Charlie has agreed to stay on the line for another half an hour or so. If you want to really kind of dig into some of the biology or have additional questions, go ahead and put those in the chat. But thank you, everyone, for participating.
All right, next question.
So Charlie, I'll sort of kick off this part with sort of a more open-ended question. I have two. The first is, I was intrigued with the ChIP-seq and enhancer profiling, and one of the things that kind of jumps out when you stare at a lot of enhancers in developing tissues is H3K27 trimethyl marks enhancers that are either going to be turned on at some point or were recently on and have been turned off. But the alternative is if you don't have that mark at all, you tend to get DNA hypermethylation and maybe a little H3K9 trimethylation, more of a permanent epigenetic repressive state. Have you guys looked at DNA methylation at all? I'm just sort of curious what that might look like.
Yeah, so great question, Mike. So actually a decade ago when we were thinking, quote, this is epigenetic, what's going on with DNA methylation? And we took a look, we knocked it off, we sent off our cells to a lab that was expert in looking at this. And it didn't seem to make any sense. And it was sort of head scratching, and we didn't seem to see something that was specific. And so when we were in Boston, we walked over and we found Alex Meisner. We redid this experiment a number of years later. And this was before we really understood enhancers very well. So this was still a number of years ago. But we found only very specific places that we were seeing changes in DNA methylation and didn't quite know what to make heads or tails of it at that point. But it turns out in hindsight, Mike, that those were in fact enhancers. And so we did see some change in DNA methylation that would seem to be specifically located in enhancers. And I think your question becomes a fantastic one of really what is the integrated model here for between heterochromatin, between polycomb complexes, between switch-NIF complexes, between DNA methylation. Really, how is this being integrated together? We're seeing fairly broad effects, but it's still, I think, a very much open question.
Yeah, and if you look broadly across diverse pediatric solid tumors from completely different lineages, right, osteo, neuro, rhabdo, you can find these beautiful developmental enhancers that are active in one and repressed in all the others. And the mechanism of repression is almost always polycomb repression. I'm just wondering if this just kind of screws up the whole wiring, and the DNA methylation might be a key piece.
Go ahead.
Okay, questions. So several ones coming in. Let's see. Have you looked at the cellular invasion or other aspects of cell metastasis in any of these models or the patient tumors?
We've looked just a little bit, and the cells, if we knock out, we looked particularly in the ARID1A model, and if we knock out ARID1A, indeed, we do see increased migration of the cells, but we didn't work it up in any really sophisticated way to understand what that was doing. And it felt fairly far away from our primary mechanistic of enhancer and chromatin changes, so we haven't worked that up further.
What do you think about the role of HDAC inhibitors in this disease?
Yeah, so it's an interesting question. When I was first starting out, and that was the question I was getting a lot because that was the only thing that was known to manipulate the genome. And so we had taken a look a number of years ago, saw some, we should probably go back, some paradoxical effects. And so we do know that in the rhabdoid tumor cell lines that the H3K27 acetyl, at least, levels are low. And so are they particularly vulnerable? When we ran things in the lab, we didn't have quite as good a motivation there for the specificity experiments. We didn't see what we thought were particularly strong vulnerabilities there. And so just sort of stopped that, and we haven't gone back to look at it since once we really know the mechanism. So a little bit, didn't see a whole lot.
Yeah, those are tough because they're so potent across, diverse cancer cell lines. So teasing out something specific is going to be really, really tough with those drugs.
So that's exactly, we had control cell lines in there and we couldn't differentiate the rabbits from the control cell line. So just screech stopped at that point. I'm worried about it being a rabbit hole.
So, back to the cell of origin question, right? This is a real, mystery, especially from a developmental perspective. You have the mouse models that you can control and activation with different Cree drivers, but have you guys considered or maybe even started to maybe play with human embryonic stem cells and do sort of organoids or embryoid bodies, something that's not necessarily biased, but just see what happens. And you could make a heterozygous and then using CRISPR to sort of in a mosaic pattern introduce the second hit and kind of see which lineages give you tumors in some sort of ex vivo system.
So I'm going to both answer that, and I realize I may not have fully nailed the 30-minute question about what genes really are responsible. And so let me give a two-part answer to this, Mike. So we took kidney rhabdoid tumor cell line, a brain rhabdoid tumor cell line, and a liver rhabdoid tumor cell line, and we turned SMARCB1 back on, and then we were looking for the Venn diagram overlap of what genes are involved, thinking, aha, we'll find the whatever, we'll find it. There was nothing left in the center that made any sense whatsoever. And really what made sense was the different lineages that were there. And so to the questioner, what the answer was, we don't think it is a specific oncogenic pathway that's there. We think this is simply developmental regulation, and this complex is not selected for per se as a tumor suppressor. It's selected to facilitate differentiation. And when that process gets broken, it's what the cancer is. So going then into your question about the embryonic stem cells. So a number of years ago, we did start working with ES cells, and homozygous inactivation is embryonic lethal. We haven't, I have a postdoc in the lab, very talented Jushin, who's been working with ES cells as a model, but we haven't done what you're asking about of looking at particular tracts and lineages. It's very hard, in one case, we have, areas of the body that it's arising from, but it turns out to be very difficult to figure out what that cell is. And so it's just something we haven't gone after.
Okay. General question. Do you find a tendency for cancers with low mutation rates to more often have disruptions in chromatin or transcription factors?
Yeah, so great question. And so I think that's really what the paradigm has become. The cancers that are driven by a signature transcription factor event, a translocation that drives a transcription factor, things like this have a tendency to have very simple genomes, or much more left shift on that pathway that I showed. Jingwei Zhang here, other people at St. Jude with the Pediatric Cancer Genome Project have done some really interesting data looking at this. But that's it, that when there are signature transcriptional events, that's what's going on. So, to me, that really leads to something that to me has been a lesson for several years now. We think of scrambled genomes because that's what we see in cancer. That's what they saw in the 1800s looking under the microscope. And so I don't want to make the mistake of saying that that's what cancer is. That is a strong correlation with cancer. And so I'm increasingly of the belief that one way to get aberrant transcription is indeed to scramble the genome, to treat with radiation, to have aging, to have catastrophic events, and then under selective pressure you can get it. But if you can actually get that altered transcription through a much cleaner hit, through an altered transcription factor, then you may not need all of this instability. And going back to that simple model that I was talking about now becomes very attractive to me in terms of thinking about what cancer's fundamental nature is.
Yeah, I agree with that, Charlie. I mean, we've seen that over and over in retinoblastoma where, you really don't need those secondary genomic lesions at all. Some of them can give a little bit of benefit, but they're not absolutely required. So
it really upends the model of what's been taught for so long about what the nature of cancer is. And I think it's just fascinating that in the exact wrong cell of origin, and Mike, you've done such beautiful work in the retina with this, just don't turn this particular transcription factor, this transcription regulator on aberrantly in this particular cell type at this particular time. And if you get all of those wrong, you pick the wrong gene, wrong time, wrong cell type, or right, then you can get cancer, and the math becomes quite simple.
And then is there any, I know it's going to be hard to answer this question, but we'll go ahead and ask it anyway. Are there any germline variants that may play a role, like through the really beautiful studies done by Les and Jingwei, but you would need a decent number of rhabdoid tumors, right, to really tease that out?
So there's germline mutations of SWTCHNIF itself. So SMARCB1 mutations in rhabdoid tumors, about 25% of those are germline. SMARCA4 in small cell ovarian cancer hypercalcemic type. This is an ovarian cancer that teenagers and young adults get that has germline SMARCA4 mutations. In terms of cooperating, I'm not aware of anything germline that's been found at high penetrance, certainly for rhabdoid tumors. So I don't know of anything else that I can think of that's right there that's germline co-selected.
So no unusual families where there's looks like this disease, but they don't have the SMARC mutation.
No. So within rhabdoid tumors, I think it's about 96% have a SMARCB1 mutation, and interestingly, the other 4% have a SMARCA4 mutation.
Got it.
So what's also mechanistically interesting, though, in the SMARCB1 mutant ones, SMARCA4 is essential. So if you've got SMARCB1 mutant and you take out SMARCA4, they can't live. But if they have SMARCA4 mutant alone with SMARCB1 intact, they can form cancer.
That's beautiful. A quick sort of broad question, open-ended question is, you've shown beautifully how the dependency screens, can really lead to fundamental insights into biology, identify, whole new complexes, therapeutic vulnerabilities potentially. What do you see kind of as the next step in that work? Hopefully expanding pediatric cancer, right, and continuing that. But where else?
So it's been fantastic. What actually drove us, I'll take maybe just a couple minutes on this because I think it's very relevant. This stuff is publicly available. It's continuing to be publicly available. We're investing in it more. We've been thinking about how to partner more here at St. Jude, and I think there's real power here. And so the problem we were facing is working up any one mutation takes a long time to power through the mechanism. We got very fortunate at figuring out the polycomb, but it's a long time. Can we just go agnostically and find these things and jump on them? And so what we found is I've actually learned perhaps even more about mechanism than I have I've learned about therapeutic vulnerability with those publications I've seen, but we've found things about mechanism of stuff I wasn't even talking about today. And so this technology of going agnostically and doing it is really powerful, but it's only really powerful in a very specific context, and that's when it's done robustly. And so when our numbers were fairly small to begin with, going genome-wide in a CRISPR screen is really a disaster. If you have 2 cell lines or 4 cell lines and you do this, the multiple hypothesis correction will kill you. And so the initial things that came out of them were false positives. But now once we got up to a robust number, now we're seeing good confidence around the things that we're finding. And so going forward with those, what we're bringing in is the drug screening to go with it. And so intersecting drug screening with the genetic dependencies is one of the things we're thinking about. Expanding to other types of pediatric cancer can be important. And now when we have, potentially to do matched cell line mutants in the background of having hundreds of cancer cell lines, we have the potential to begin to understand the contributions of specific mutation types as well.
Fantastic. All right, and then I guess that is the last of the questions. And for those who have stuck around, just wanted to put in a quick plug for the Cancer Center and also the solid tumor that if there are researchers out there that study rhabdoid tumors, we do have a handful of orthotopic xenografts that we distribute free of charge to any researchers who want to dig deeper into some of this biology. So thank you again.
I should mention the St. Jude Cloud. If you
yes.
go to St. Jude Cloud, all of this stuff, in fact, Mike has been the major champion of this, or a major champion of this. If you just go St. Jude Cloud, you can find our resources there. Mike has set up the Childhood Solid Tumor Network. All of these incredible mice and prior samples are there. There's brain tumor samples that are coming. I think the leukemia samples, I can't remember if they're there yet. Will be there shortly.
Again, thank you so much, Charlie. I thought the discussion was fantastic. Terrific questions. It was really a wonderful.
Looks like Madan has a question there, Mike.
Oh wait, fantastic. You had a few other hits, and from the drug screening. And do you have sort of general idea about the compounds and how they might mediate their effect? Have you followed up on any of those drug hits? Basically?
Yes. So, for instance, the amacitaxine, we did a CRISPR screen that was 100% on target. When we looked at, so the CTD squared was not designed to be part of the pediatric cancer genome project, so they weren't linked together. But we crossed over and took a look, and so the receptor tyrosine kinases, the drugs that were scoring well in the CRISPR space, although we were then finding out that some of the receptor tyrosine kinases were a bit dirty as well. So there really does seem to be power between the intersection of the genetics with the drug space. in terms of being informative. And we have a number of other ones that are coming along, just some fascinating mechanistic stuff, some hits out of the, we've gotten some hits out of the screen that we just never would have predicted mechanistically. But as soon as we begin to look at enhancer function, they're right on. And so we're finding new regulators of enhancer function, new regulators of chromatin regulation that are quite interesting.
Quick question about development and switch SNF regulation. So it says a lot of the work has been done with the loss of the core components in the developmental literature, and it seems like PBAF and BAF tend to have very different contributions to different developmental lineages. So how do you explain this given the model is that the complexes cooperate?
Yeah, so it's really an area of interest, and I would go back to one of the very first slides I was showing with all of those variant subunits, right? So there's 4 different BAF45s, there's 3 different BAF60s, and this combinatorial complexity is really immense. And so what we have is 3 families that have pretty signature subunits as part of them, but within those families, there's still quite a bit of diversity there. And so we don't really know what that diversity is about. Is it about recognizing specific transcription factors? Is it about recognizing specific chromatin modifications? Is it about recognizing specific co-adapter proteins and other interactors? And is this really part of bringing the specificity? When I go back to my graduate work when I was with Stan Korsmeyer and we found a Hox gene that was involved in leukemia, kept looking for this binding site. And one of the things that was really frustrating was there's just not enough information in a Hox gene's DNA interaction domain to actually give you genomic specificity. You can work out some sequences, but it's not enough. And so what we think is beginning to happen is it may be that transcription factors are interacting with the switch-NIF complex to bring that added specificity. And so it's a little bit hand-waving on my part because we haven't been able to mechanistically nail it yet, that's what underlies it, but I think it's a pretty likely bet that it's that degree of diversity that is bringing up where that specificity is coming from.
And I know you haven't had time to really get into this, and it may be impossible with current systems, but if you look at developing tissues and look at the changing, enhancer landscape and epigenetic landscape, the thing that really jumps out at you is how incredibly fast and dynamic these things are happening. In the course of a day or 2 days in developing retina or cerebellum, the number of enhancers and promoters that are turning on and off is just mind-blowing. So you could imagine then that also the coordination and kinetics of these different processes, right, with this complex, has got to be nailed exactly, right, to get it right. I don't know how you would study it, but it's a
one of the, so that's exactly right. And Mike, so all of the things that I showed today were essentially steady state experiments. We delete it, wait, 48, 96, 120,
yeah.
And see what happens, or we add it back and do the same thing. So precisely because of this question, we've begun to work with the nuclear hormone receptors, where you can turn on expression, boom, and turn it on and turn it off. And these things are known to interact with the switch-NIF complex. And so now using these as tools and the diseases that are associated with them as tools to begin to understand this rapid response signaling and really what is happening there, because it's a really important question that I think is going to go right back to your question, Mike, about what is cell of origin. It's going to be cell of origin and the right context as well. For instance, we found that memory T cells, if they didn't encounter antigen, would not, in other words, if we knocked out the class 1 interaction, they could not, the CD8 cells could not interact, could not turn into cancer. So they needed this exogenous signal. And so it might not only be cell of origin, but cell of origin under the right signaling context as well.
Yeah, and sort of a separate plug for the human, maybe a stem cell or some other type of model is human development goes a lot slower. So it gives you a better chance to catch that moment in time than mouse development is so rapid in the mouse. And some of these organoid systems that you can do with stem cells now, you can kind of stretch that out, right? And really try to capture those key events and moments if you guys do end up developing that. So one case where humans a little easier maybe than the mouse in that context.
Maybe this can be one of our formats that we actually launch new grants through, Mike.
I'm ready to queue it up.
A big Q&A online. We find a project together. Yeah,
well, it would complement really, RB beautifully, right? Sort of second hit.
It's really interesting to think about. Yeah.
Oh, fantastic. All right, I think that is it. Thank you so much for sticking around and engaging in this really fantastic discussion. I think that's probably the highlight for many of us. So thank you, Charlie. And thank you all participants for joining us and tune in next time. Thanks.
Thanks all. Take care, everyone.